Welcome to AP Biology. This video is on adaptive immunity, or the specific immune system. In adaptive immunity, we have white blood cells called lymphocytes. These lymphocytes are extremely specific for the foreign invaders that they target. This is what makes adaptive immunity different from innate immunity. It's going to be the specificity of the white blood cells involved, the lymphocytes. There are two types of lymphocytes. You have B cells and you have T cells. Both come from stem cells within the bone marrow, but they mature in different places within the body. The B cells mature in the bone, which is why they're called B cells, and the T cells mature in the thymus gland. Once these lymphocytes have matured, they are going to be stored within the lymphatic system. The foreign invaders that the B and T cells respond to are called antigens. An antigen is a foreign molecule that elicits an immune system response. It could be a virus, it could be a bacteria, it could be pollen. The important thing here is that B and T cells are both specific, extremely specific for the antigens that they target. B cells are going to play a large role in long-term immunity because they are going to create proteins called antibodies. Antibodies remain in your bloodstream for the duration of your life and protect you against an antigen if you are ever exposed to it in the future. T cells do not lead to the production of antibodies. They are going to be more involved in the immediate immune system response. B and T cells recognize antigens based upon structures on the outside called epitopes. The part of the lymphocyte, the B or T cell, that responds to the epitope of the antigen is called the antigen receptor. The antigen receptor is a protein on the cell membrane of the lymphocyte. We literally have thousands of different lymphocytes within our bodies, all containing different shaped receptors. We accomplish this by going through extensive amounts of genetic recombination. When an antigen enters your body, there is going to be a lymphocyte that has a receptor for the epitope of that antigen. In this case, it's this lymphocyte right here. Once this lymphocyte has detected the antigen within the body, once the antigen has bound to the antigen receptor, the body is going to make copies of this specific lymphocyte. We call this process clonal selection. You're making clones of this particular type of lymphocyte. Two kinds of clones are made. One kind called the effector cell. Effector cells are short-lived. They are used immediately within the immune system. Memory cells are more long-lived. They remain in your bloodstream for weeks or months and are going to protect you if you have a second exposure to that antigen. This process of clonal selection is going to take place within your primary immune system response, your first exposure to an antigen. Your secondary immune response takes place if you encounter that antigen in the future, and that's going to involve the use of antibodies to kill that antigen. The particular type of B cell that produces these antibodies is called a plasma cell. A plasma cell is an effector B cell. Here we see all the different types of lymphocytes that can be created. Number one, again, you have the effector Bs, which we call the plasma cells, that produce antibodies. You also have memory Bs, which will remain in your bloodstream for weeks or months, that can become effector Bs at a later time. There are multiple types of effector T cells. One type is called the cytotoxic T, and that's going to be the lymphocyte that really attacks the antigen. It actually doesn't attack the antigen itself. It attacks any antigen-infected cells within the body and kills them. Helper T cells are considered the core of the immune system and are arguably the most important lymphocyte within the immune system. These helper Ts we're going to see are going to cause the production of both plasma cells, which are going to create antibodies, and cytotoxic Ts, which are going to kill off any infected cells within the body. Finally, the last type of T is going to be the memory T, which again remains in your bloodstream in the case of a recent second exposure. How can your body tell what belongs inside of it and what doesn't belong inside of it? We have the ability to distinguish self versus non-self. To do this, we have these big billboards on our cell membrane that designate your cells as being a part of you. We call these billboards MHCs, major histocompatibility complexes, and they're made up of glycoproteins. There's two types of MHCs. One is called a class 1. Class 1 are found on all your body's normal cells. So this class 1 MHC is really how we're able to tell what normal cells belong inside of us and what don't. Class 2 MHCs are found on many of our white blood cells. In particular, we're going to see them located on macrophages. Macrophages are these large phagocytic white blood cells which are part of innate immunity or the non-specific defense. The MHCs are going to perform two functions within the body. One is in self-recognition. Like I said, they're going to be a billboard that allows our immune system to detect what cells belong inside of us and what doesn't. The second function it's going to perform is called antigen presentation. Antigen presentation takes place in order to present the antigen to a member of the T lymphocytes. If a normal body cell is infected by an antigen, it'll take a piece of that antigen and hold it out, and it will present that antigen to a cytotoxic T cell. So if you look at this picture here, this cell right here is the antigen presenting cell. Let's say it's a normal cell within the body. It gets infected by a bacteria. It's going to use its MHC, which is going to be a class 1 MHC if it's a normal body cell, to hold out a piece of that antigen. And it presents that antigen to the cytotoxic T cell. When the cytotoxic T cell responds to that antigen, it's going to respond by killing the infected cell. So cytotoxic T cells respond to a class 1 MHC. If a macrophage engulfs an antigen, if it engulfs a bacteria, it will also hold out a piece of that bacteria. It'll also become an antigen presenting cell. These white blood cells, like the macrophages, have a class 2 MHC, and they use that class 2 MHC to hold out a piece of the bacteria, a piece of the antigen, and they will present that antigen to the helper T cell. So helper T cells respond to class 2 MHCs. Once that helper T cell has been activated, like I said earlier, it is then going to activate both cytotoxic T cells and B cells, which are going to become plasma cells. So helper T cells respond to the class 2 MHC. Here we see an overview of how the immune system is going to work. And you can see that once an antigen enters your body, it can go in 3 different directions. The most important direction to understand is the direction right down the middle. Chances are, if an antigen enters your body, it is first going to be engulfed by a macrophage. It's going to be detected within your innate immunity, your non-specific immune system. When that macrophage engulfs the antigen, the macrophage becomes an antigen-presenting cell. It's going to hold out a piece of the antigen using its class II MHC. And we just said that the macrophage is going to present the antigen to a helper T cell. Here you can see why we consider the helper T cell the core of the immune system. The helper T cell is going to stimulate the production of cytotoxic Ts, and it's going to stimulate the production of B cells. The cytotoxic T cells go out and kill any infected cells within the body. The B cells turn into plasma cells, which are going to make antibodies. Again, these antibodies are going to remain in your bloodstream in case you have a second exposure later on in life. You can see here that memory Bs are also made, memory Ts are also made as well. Here you see the route most often taken when an antigen enters your body. If the antigen infects a body cell, that infected body cell can also directly activate the cytotoxic T. We know that the cytotoxic T responds to the class I MHC that the normal body cell contains. B cells do not respond to MHCs. However, the B cells can be activated directly by the antigen. If you look at this flow chart here, you can see it's divided up into two sides. The side in which the antigen is immediately combated using the cytotoxic T cells is what we call cell-mediated immunity. This long-term immunity in which antibodies are produced is called humoral immunity. Let's look at both in more depth. We begin with the cell-mediated immune response, and this is going to involve the T lymphocytes. Here we see the two types. We have the helper T, and then we're going to look at the cytotoxic Ts. We said that helper Ts are the core of the immune system, so they actually function in both humoral immunity and cell-mediated immunity. It's going to activate both sides of the flow chart. We know that helper Ts respond to a class II MHC. Here you see on the bottom right here a macrophage presenting the antigen to the helper T, and it's going to present that antigen using its class II MHC. There is a protein that is on the surface of the helper T called the CD4 protein, and that is going to enhance the affinity between these two cells. I think of it working kind of as a vice that holds these two cells together during the process of antigen presentation. Once the macrophage has presented to the helper T is going to respond by releasing a chemical messenger called the cytokinin. The cytokinin is going to be the chemical messenger that stimulates the production of cytotoxic Ts and stimulates the production of the B cells. It's the chemical messenger that makes that happen. When the B cells have been stimulated, a chemical is going to be released called interleukin-2. Interleukin-2 is going to be the chemical messenger that causes the B cell to differentiate into a plasma cell, which is then going to make the antibodies. This helper T cell is the cell that gets infected by the HIV virus, which is what makes the HIV virus such a deadly virus. It effectively takes out the core of the immune system. Cytotoxic T cells, or killer T cells as we call them, we say that they respond to a class 1 MHC, so they respond to normal body cells that have been infected. They have a protein called the CD8 protein. So when a normal infected cell becomes an antigen presenting cell, it's going to present the antigen to the cytotoxic T. There is a protein that's going to work like a vice here, but we call this the CD8 protein. When the antigen has been presented to the cytotoxic T cell, the cytotoxic T cell kills the target cell by releasing a protein called perforin. Perforin forms a pore, a hole on the target cell membrane that's going to cause the target cell to lyse, to burst. Cytotoxic T cells also respond to cancer cells within the body. They also respond to tumor antigens. Humoral immunity. Within humoral immunity, B cells are going to become plasma cells, which create antibodies. An antibody is a quaternary protein. It's a protein made up of 4 polypeptide subunits. The antibody, which is also called an immunoglobulin, IgS, has 4 subunits, 1, 2, 3, 4, and they create this Y-shaped protein. 2 of the subunits are called light chains, and 2 of the subunits are called heavy chains. And where the light chain and the heavy chain meet, we have our antigen binding site. So each antibody, or each immunoglobulin, is going to contain 2 antigen binding sites. Once the antibody has bound to the antigen, it can do 2 things. Number 1, it can neutralize it. They literally cover the entire surface of the virus or the bacteria, which prevents them from infecting other body cells. And it's going to work as a signal that's going to cause a local macrophage to come and engulf that antigen. The other thing that can happen is the antibody can form a complex called a MAC, a membrane attack complex, in which the antibody is actually going to form a pore and is going to lyse or explode the bacteria. There are 2 different ways in which we acquire antibodies. One is through active immunity. That's your primary and secondary immune response when you are exposed to an antigen. The other form is called passive immunity. You're not exposed to the antigen. You actually get antibodies from your mother. These antibodies are found within breast milk. So the newborn baby is resistant to any antigen that the mother is resistant to. We can categorize antigens based upon how they lead to the production of antibodies within the body. There are 2 types of antigens. One is called a T-dependent antigen. A T-dependent antigen is an antigen that leads to the production of antibodies through the helper T cell. A T-independent antigen is an antigen that directly stimulates the B cells, which then make the antibodies. So they're T-dependent or T-independent depending upon whether or not they first go through that helper T cell. Vaccinations are performed to build up antibodies within the body. When someone receives a vaccination, they are injected with an inactivated bacterial toxin, a killed pathogen, viral proteins, viral genes, a piece of an antigen. And when a piece of the antigen is brought into the body is going to have an immune system response and create antibodies for that antigen. So even though you're not directly exposed to the actual antigen, you will have built up antibodies if you ever encounter that antigen within the future. Finally, there are several different disorders of the immune system. One disorder is an autoimmune disorder. This is when your body fails to recognize self. You fail to recognize these MHCs. There are different types of autoimmune disorders depending upon what part of the body is no longer recognized. One disorder is called lupus, and this is when the body targets the histones in DNA. Rheumatoid arthritis is when the body or the immune system targets the cartilage and bones within joints. Multiple sclerosis is when the immune system targets the central nervous system, in particular the Schwann cells on the axons of neurons. Finally, we have type 1 diabetes, diabetes mellitus, in which the immune system targets the beta cells of the pancreas. There are also immunodeficiency disorders. This is where you have a defective or an absent immune system. The first one is called SCID, S-C-I-D, severe combined immunodeficiency. This is when an individual is born with an extremely weak or defective or totally absent immune system. Hodgkin's disease is caused by a cancer that targets the immune system. And AIDS is caused by the HIV virus, targeting the helper T cells within the immune system, which makes the immune system dramatically reduced. This concludes our video on the immune system.